Hello, my name is Agata Błaszczyk. This is a photo guide showing how to assess the activity of the tested sample on the saltwater crustacean of the genus Artemia. In this essay, we can examine sample's impact on the tested species. The scheme shows subsequent stages of the sample's preparation. It's important that the samples are transparent and free of any dirt, so that the digestive system of crustaceans doesn't clog. And also, please pay attention to the sample's viscosity. It's most convenient to use ready kits to assess sample's activity. Why? The commercially available sets are user-friendly, they save our time, and what is also significant, they are available in different variants regarding the test organism: bacteria, ciliates, rotifers, crustaceans, microalgae, and higher plants. Such biological activity assessment screening tests have been developed by a research team led by Professor Pierson at the University of Ghent from Belgium. In the case of small crustaceans test, the biological activity of the tested sample is evaluated in multi-well plates. This assay lasts 24 hours. After this time, Artemia mortality is measured. It should be mentioned that this bioassay can also indicate compounds accumulation and the behavioral changes of crustaceans. Commercially available sets include all the needed equipment to conduct the tests: 24 well plates, micropipettes, Petri dishes, parafilm, vials with cysts, and components to prepare the standard seawater. You would need to provide basic laboratory equipment: small laboratory glass, automatic pipette, microscope, and incubator. The first step is to prepare the standard seawater. To do this, pour the content of vial number 1 into a flask with 800 ml of distilled water. Shake until all the salt is dissolved. Then pour the contents of the remaining 6 vials into the flask. Remember to stick to the given order. Then, fill up the flask with distilled water to 1000 ml and shake it afterwards. What's important, if you want to test brackish water samples, you would have to prepare a standard seawater with reduced salinity. Brackish water must also be used as a dilution fluid for the preparation of samples. Now, let me guide you through the test procedure. First, the vial containing the cysts must be completely emptied into one of the Petri dishes. Make sure that all cysts have been rinsed out of the vial. Then, add 10 ml of standard seawater to the Petri dish and mix it gently. Cover the plate and incubate it in 25 degrees for 30 hours in continuous light. In such conditions, hatching should start after circa 20 hours. After 30 hours, most larvae should have molted into the instar 2-3 stage. In the meantime, prepare serial dilutions of your samples. Bear in mind that the final volume of each sample should be 5 ml. Is there any special method of filling the test wells? Every single column is meant to be occupied by one sample only. It means that each solution has to be transferred to all the wells of the column. Test wells are marked horizontally from A to D and vertically from 1 to 6. What's important, wells in row D are rinsing wells. Start distributing the test solution from the control wells to the well with highest concentrations. When all the wells are filled with adequate solutions, transfer the larvae to the wells. This is a two-step process. Step 1. Transfer about 50 larvae from the petri dish to each rising well. Use the microscope to assess the development stage of each individual. Step 2. Transfer exactly 10 larvae from the de-rinsing wells to the 3 remaining wells in rows A, B, and C. Start from row 1 and proceed till row 6. When the process of setting the test is completed, incubate the plate in 25 degrees for 24 hours in the dark. Remember, the plates should be covered with parafilm. How to read the test results? After 24 hours of incubation, check the wells in rows A, B, and C and record the number of dead and living larvae. Larvae are considered dead when they do not show any movement during 10 seconds of observation. Record the results in the table. Score the total number of dead organisms for each concentration and afterwards calculate the percent of mortality. Nonetheless, if the mortality in the control wells exceeds 10%, the determination is considered invalid and must be repeated. The last stage of the test would be assessing the LC50 value. There are many procedures for calculating this parameter. In this test, we use a very simple graphical method. First, we should mark the concentration or dilutions of the test solution on the y-axis. Next, mark the calculated mortality percentage for the given concentration on the x-axis. Then, connect the points with the straight line and extrapolate the LC50 value. Thank you for your attention.